we now move on to Dr. Mark Ware. He's the Director of Clinical Research at the Alan Edwards Pain Management Unit at McGill University, an Associate Professor in the Department of Family Medicine and Anesthesia. He's also the Executive Director of the Canadian Consortium for Investigation of Cannabinoids and the recipient, the lead on a team who received a 2015 ALS Canada Clinical Management Grant. Welcome, Dr. Ware.
Thank you very much. Good afternoon, everyone. I hope you can all hear me okay, and I am going to now switch over to my screen, and hopefully you can see that. I will bring up the presentation. And David, is that showing up online okay?
It is, yes.
Fantastic. So thanks, and again, thank you for the invitation. I don't think I have to remind anybody that cannabis is kind of the story of the year with the federal government's proposals to legalize and regulate cannabis, which will probably start to hit everybody's neighborhoods sometime next year and in the years to come. So cannabis very much a subject of interest across Canada and, in fact, increasingly globally. I've been involved in doing cannabis research for medical purposes for close to 18 years now, mostly in chronic pain management. And it's with that context that I've been very fortunate to work with colleagues Angela Genge, Colleen O'Connell, and others in the ALS community to consider whether cannabinoids may have a role to play in treating patients with ALS. And so what I'd like to do in the next 15 to 20 minutes or so is give you a very quick overview as to why we think this is actually a credible thing to study, and then some very short outlines as to some of the challenges that we face in making studies of this kind actually happen. What's important to realize is that we know in a number of different clinical contexts that people self-medicate with marijuana or cannabis. And just for the record, cannabis and marijuana really are the same thing. Marijuana is a slang name for cannabis. It has tons of other names, weed, pot, whatever people call it. But it's like cannabis is the scientific name for the plant that is grown and produces the cannabinoids that we are interested in. But people use cannabis as a self-medication for a huge number of different conditions. We've done surveys of patients with HIV, AIDS, epilepsy, non-cancer pain, MS. Anywhere from 15 to 40% of patients will actually, when asked, report that they've tried using or have used or currently use cannabis as part of their therapy. And often this goes undisclosed to physicians. It's sometimes done kind of quietly. The drug is very stigmatized. It's very smelly, and people tend to kind of hide this away. But it's quite clear that patients report using it to improve pain. Is the thing that has obviously interested me the most, but it's quite interesting to see that appetite stimulation, seizure control, spasticity, sleep, mood improvements, and so on all get reported in these patient surveys. And this is true in ALS as well, and it's intriguing to see that in a survey done over 10 years ago now of patients who use cannabis reporting help with things like weakness, speech difficulties, hypersalivation, difficulty swallowing, breathing, as well as pain, spasticity, the appetite, some of the more classical issues that have been seen in other conditions. And it's an intriguing drug that can appear to affect this many different symptoms, essentially using a single therapy. And so what I'd like to try and do is tease out how this is possible for a drug to somehow target this many potential interactions. And I guess to do that, we need to look at what cannabis is, and to first realize that when people talk about medical marijuana or cannabis as a therapy or legalizing cannabis, we're talking about a very complicated plant which contains a large number of ingredients. The two that we've focused on historically the most are THC, tetrahydrocannabinol, and CBD, cannabidiol. And as you can see from this picture, the two molecules are actually very similar in their shape and their structure. There's a tiny change in one molecule from THC into CBD, but that really transforms the effect that particular drug has in the human body. And THC is the drug that we perhaps know most about. It's the psychoactive ingredient in cannabis. It's what gives it its euphoric effects, as well as a lot of the effects that have been studied medicinally, as you'll see in a few moments. But CBD, cannabidiol, is equally important now and is being studied for patients and in fact, young people with epilepsy. There was a recent study done and published in the New England Journal suggesting that CBD could be effective in treating seizures in children with very severe refractory seizure disorders called Dravet syndrome. But the cannabis plant produces both of these, and in fact it produces hundreds of other cannabinoid type compounds, similar molecules but with tiny changes in the structure. And in fact these two compounds come from forms of cannabis that can be very closely related but also very different. The marijuana type that's used to make THC is in some ways a very similar relative to the cannabis that's used to make hemp, or the ropes and paper and the hemp oil that you see in the body shop, but which of course doesn't contain any cannabinoids, but the flower will express the CBD compound. So we have to realize that cannabis contains these two major ingredients among others. And they come off, and if you look really closely at the flower of the cannabis plant, when it reaches its flowering, it produces these tiny little sort of golf ball type structures on the flowers, and inside these little bags are the cannabinoid compounds. And what people do when they're self-medicating, and what scientists do when they're trying to study these compounds, is to remove these little tiny trichome structures from the flower and isolate them into pure compounds or into extracts or oils, which can then be used in clinical research. So there's an act of separating these compounds away from the plant which has to happen, and then they need to be somewhat modified in order for them to have therapeutic action. So the raw flower, if you just chewed on a raw cannabis flower, you wouldn't get much of an effect because those cannabinoids need to be modified, and the way you do that is through heat. Heating a cannabinoid releases a special carbon dioxide molecule from the molecule, which then activates it, makes these compounds active in the plant, which of course it makes sense when you consider people using cannabis tend to do something in form of heat. They either bake it or cook it, or they heat it and smoke it or vaporize it using a heating element, and that heat is actually a very important activation of the compound itself. So you have to remove the compounds and then heat them, and then you get active THC, CBD, and then a dozen of other compounds. And some of these have been studied in preclinical studies in animal models. Some of them are emerging now into early phase clinical studies, but CBD we're studying quite a lot, but these other compounds, CBG, CBD, the variants, the acids, these are all compounds, and this is just a summary of the ingredients that we're also looking at. And then if that's not complicated enough, cannabis contains other compounds which are the ones that give it its smell. And when you smell weed walking down the street or in your basement from your teenage sons, you are smelling these other compounds: myrcene, limonene, linalool, beta-caryophyllene, all of which seem to also have some degree of therapeutic activity by themselves if you isolate them and look at them in different models. And it's thought that these ingredients also contribute to some of the effects, and maybe one of the reasons, and we don't know this yet, but it's been hypothesized that the reasons why people like certain strains of cannabis over others has not just to do with the THC and CBD levels, but the presence of these other ingredients which give it its aroma and its taste and potentially some of its pharmacological activity. So we're dealing with a very complex mixture of chemicals. When somebody's inhaling or ingesting a cannabis oil or product, they are ingesting a large number of compounds, which gives it its complexity, but also makes it very challenging to study. As you've been hearing about earlier, clinical research tends to use single compounds, single molecules, and placebo-controlled trials where you either have the molecule or a placebo get very challenging when you're dealing with a compound that contains, or a plant that contains multiple different ingredients. We know best THC. It's the one that we've been studying. It's been identified for about 50 years now, and it's been well studied in a lot of different animal and human conditions. We think that the way THC works is it binds to a specific receptor that we all have in our bodies, in our brains, in our spinal cords, all the way out into the peripheral tissues. And in fact, these cannabinoid receptors and compounds are found throughout the body and play an enormous role in a number of different physiological processes. But the receptor itself appears to be responsible for, at least in the nervous system, for sort of fine-tuning the way nerves talk to one another. So as synapses release transmitters, the activation of the second neuron releases endogenous cannabis molecules, so molecules that our body makes that mimic THC, which then bind to these receptors and smooth out the process. So it's a feedback loop mechanism. It's happening in all of us all the time, and by administering a cannabis extract from outside, we're actually hitting these receptors and affecting the way our nerves talk to one another. And this, of course, happens in a huge number of different areas of the brain. The CB1 receptor is probably one of, if not the most widely expressed brain receptor that we have. And it affects movement and memory and appetite and pain and muscle control, emotions, and so on. And so by administering cannabinoids, we're hitting a large number of these important brain areas, but we're also targeting other parts of the body, from the heart to the GI tract and so on. And this helps us understand how it is that cannabinoids, THC in particular, but the other ingredients as well, can affect so many different systems, as you saw from the slide suggesting why patients with ALS may try self-medicating with cannabis. It appears to be a target that's distributed throughout the body and brain and gives rise to such a wide variety of different effects. This is a very simplified diagram. Believe it or not, the mechanism by which the cannabinoids binding to the receptor change what happens inside a cell, whether it be an immune cell or a nerve cell or others, is incredibly complex. But it's being worked out now, and it's being clearly identified that these cannabinoid receptors are very important in our normal physiological activity, and so we're beginning to target them now as potential therapeutic agents for treating conditions where this system may have become dysregulated. And just by way of summary, in the last 10 years or so, we've recognized that in ALS itself, the endocannabinoid system, or the ECS, whereby these receptors and these ligands that we produce may have an effect on the CB1 receptor, the CB2 receptor, a whole other range of processes that involve the cannabinoid receptors, affecting inflammatory markers, affecting the release of various activating or excitatory currents in the CNS, in the nervous system, and has been explored and have been explored in a number of animal studies of ALS, and appear to be quite important and potentially interesting therapeutic products to use in humans. So we're building up a better idea of what the rationale might be for cannabinoids working in the brain and in the body. We're beginning to look at animal models where this suggests that there might be a good reason to look at these compounds. The question, of course, is can we use them, or can we recommend or study them in humans with ALS in a formal clinical context before we make recommendations or treatment decisions with cannabis as therapy in this very important population. It's really important to know that while the talk about medical marijuana and cannabis in general is quite recent, there have been and are a number of cannabinoid compounds that have been available on prescription for some time now, including THC as an oral compound. It's been available for the treatment of nausea and vomiting in patients getting chemotherapy. It's been approved as a drug to improve appetite in patients with HIV, AIDS. We have nabilone, which is a synthetic THC-like compound. It's been approved as an antiemetic drug for chemotherapy-induced nausea and vomiting, and these drugs have been around since the mid-1980s. More recently, Sativex or nabiximols, which is a spray containing the two compounds, THC and CBD, in standardized measures. They were extracted from plants. They've been produced in a spray which you can administer into your mouth. And this has been approved for MS pain, MS spasticity, as well as for advanced cancer pain, and is available now through prescription in Canadian pharmacies. So we have access to these compounds already, and if you're interested as a patient or as a family member, you need to speak to your physicians about whether any of these could be considered in advance of thinking about whether herbal cannabis might be an option, which of course is available through legal frameworks in Canada, various state programs in the USA. There are other countries that have medical marijuana programs, but this is very challenging in the sense that there's no formal approval. These aren't medicines that can be prescribed and delivered through the pharmacy and are undergoing a number of clinical tests to establish whether there are some valid claims around them. So we need to sort of separate out what we know as pharmaceutical cannabinoids from the medical marijuana or cannabis that we are currently hearing so much about in the media. And cannabinoids have been studied, and I've just blasted through a number of different studies, and these are all been done in the last 10 years, so this is relatively recent work, looking at some of these cannabinoids like nabilone in a range of different pain conditions, the Sativex, nabiximol spray, also in a number of different conditions, including arthritis and some very severe nerve injury models, THC in MS and spinal cord injury as well, some extracts of cannabis looked at in the UK for MS patients with very severe spasticity. And then, of course, we have the herbal cannabis products ranging in THC levels because the plant expresses different levels for a number of different pain conditions, predominantly neuropathic pain where the nerve is damaged and causes very severe, very uncomfortable pain syndromes. And these studies have all been completed, and you can see again the dates for all of this work is really in the last 10 years that we've begun to see serious efforts looking at cannabis as a therapy in these different disorders. So switching back to ALS for a moment, so just to summarize that table that we looked at earlier, patients have reported, and there's rationale to think that cannabis may be effective, or cannabinoids, in the management of patients with ALS, looking specifically at things like pain and spasticity, wasting, difficulty breathing, hypersalivation, mood disorders, and so on, and especially insomnia, which appears to come back in many, many different conditions, patients reporting cannabis helping them to sleep. So we need to target all of these things if we're going to study cannabis as a therapy in ALS. There have been some very small studies looking at THC alone as a pure single agent in doses of 5 to 10 milligrams, small studies with small numbers of patients, single doses, separated out so that patients can try the drug and see what the response is. Higher doses, 10 milligram doses, a fairly deep, but big dose of THC appears to have more adverse events, and these typically are, they're not dangerous, but they're quite important, especially if people have to go about their activities or drive a car or conduct important functional activities, things like drowsiness, the feeling of high, feeling blood pressure dropping, or feeling that when you stand up you feel dizzy, drowsiness, dizziness and weakness, and so on. And these effects increase with the higher doses. So the challenge is finding what is the right dose that can give you the best beneficial effect while reducing the likelihood of having these kinds of side effects. What makes it even more challenging is that people seem to respond very differently to cannabis. Some people are very sensitive to THC effects. Other people require much larger doses to be able to report or feel any effect, either positive or negative. And this may be because of differences in the way we metabolize cannabinoids, the differences in the kind of receptors that we have in our bodies. And so this is challenging. That's why small studies are so difficult to interpret. Another study done where patients were given THC or a placebo in a crossover fashion, meaning that during one sequence they receive THC, then they wash out, and then they get placebo, and other patients receive a placebo first and then the THC afterwards. These studies did not show any major differences between THC and placebo in terms of muscle cramps, and then that was the primary outcome. And then they were also looking at things like sleep and anorexia and depression. They also didn't find effects, but they were very small doses and also very small studies. And we believe that this work needs to be reproduced. It needs to be retested, looking at potentially other outcomes, but also potentially building in other cannabinoids. This is only THC. And it may be that we have to have cannabidiol or these other compounds in the product in order to get these therapeutic effects. This is what patients are telling us, that the pure THC doesn't seem to help, but the whole cannabis extracts appear to be better. So we need to explore that in more detail. And we're lucky in Canada in that we currently have a formal federal program where there are, I think as of yesterday, 52 companies licensed to grow cannabis for medical purposes. There are hundreds of others in the queue waiting to be approved. Of course, some of these are waiting to grow cannabis for non-medical purposes, but there are a number that are trying to develop cannabis extracts and very specific products for study and research purposes in clinical trials. And we're very fortunate to have partnered with one of those and the ALS Canada to get a grant to begin to explore this more seriously. And as I mentioned earlier, the challenge is somehow picking products that have a standardized level of both THC and CBD and these other ingredients. And just looking back, this is going back a couple of years when we looked at the differences in THC and CBD levels between different cannabis plants that were available through the regulated model in Canada. And you can see a huge range of THC levels available from 15 to 20%, most of them, and that's the percentage of THC in the flower. Some of them having very high levels of CBD, others having fairly mixed levels of both THC and CBD. So the challenge is picking a series of these ratios, THC and CBD, to use in a clinical trial. A nod of thanks to my collaborators, Colleen O'Connell, Angela Genge, Andrea Benedetti, our statistician, the team at the Montreal Neurological Institute who are helping to manage the trial, and of course ALS Canada and the Green Sky Labs, the company that's producing the oils for us to use in the study. The study is ongoing. We are in the process of working with the company to get the government to approve the oil for its use in patients. And the challenge is that the company that produces this oil has to demonstrate to Health Canada that the oil is stable. It can be left in the fridge or in the shelf for a period of 3 to 6 months without losing any of its effect. And these companies that produce these oils have to put the oil through a series of very, very high quality tests to ensure that they meet the requirements of the study and also meet the safety requirements of Health Canada for them to be used in clinical trials. We're expecting the first release of these products to come through by the end of this year so that the study can begin. Once the study is underway and we're looking to explore the effects between THC and CBD on the symptoms of interest in ALS, there is intriguing information suggesting that cannabinoids may also affect actual disease progression. And this is very, very early work, but it's coming out of some animal models where the animals were given cannabinoids and it was then measuring with models of ALS and measuring their survival. And it appeared as though there may be some effect there. So these are going to take much longer term studies than the one we're intending to do, where we will be administering single doses over a period of a week and rotating through different various levels of THC and CBD, but the duration of exposure will be relatively short. And then we will need to extend, as I think you heard from one of the other studies, longer-term extension studies to see whether the dose needs to be adjusted and then ultimately whether there can be any changes in how the disease progresses. But these are things that we're well aware of and that require formal study in the future. We need to know what happens with doses. We need to know whether patients can tolerate different levels of THC, CBD. We're using an oil model in our first study where the patients will consume the oil in a capsule form. So kind of like swallowing a pill, it can be opened up in a capsule and taken into a tube if needed. But this oil allows us to standardize the amount of cannabinoids that the patient is consuming in the study. We are aware that patients use other delivery systems. They smoke or vaporize cannabis, and so that probably needs to be explored as well. Some people don't like swallowing or are unable to swallow medicines, and so an inhaled route may be an interesting avenue for further study. And of course, the final note that I'd like to make is that all of this research has been pushed and stimulated by patients who have reported that these drugs help, and of course with epilepsy and MS and HIV, there was a political push to have cannabis available and studied because they were reporting these effects. So we can learn a lot from patient experience, from what happens in the real world, and then bring that information back into our clinical trials and try to design studies to answer questions that patients are facing for important symptoms that they're living with on a daily basis. So harnessing this, what I call the living laboratory, is a really important part of this work, relying on patient exposure and patient feedback to be able to design the right kinds of studies that will be most important. With that, I'm going to stop. I realize that I've put a lot of information together for you very quickly, but just to try to hopefully identify the rationale for doing this kind of work, where we think it might work, and how we're proposing to approach this, given the complexity of both the plant itself as well as the complexity of the system, the endocannabinoid system that we're proposing to target with this research. And with that, David, I'm happy to try to entertain any questions.
Thank you very much, Dr. Ware. Really appreciate it. That's a very comprehensive overview of something I think people maybe really wanted to hear and didn't fully understand some of the biological aspects of. In order to move along with the program, we're actually going to we'll follow up with questions for you offline, if that's okay, and we'll move along. Is that?
Yeah, that's fine with me. Please send me any questions by email, and I'll do my best to get back to you. Thank you very much for giving me the opportunity to present this work.
Thank you so much for joining us today. We really appreciate it.
My pleasure.